Yeah, he does.
Test, 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 test.
It'll take a while to turn it back on.
Test, test.
Thanks.
Testing, testing. Oh, that's good.
For the signal to see if we're good. Very good. All right, so thanks everyone. This is a great turnout for a local celebrity. So it's my great pleasure to introduce Andrew Bowler, who's an assistant professor here in the Department of Chemistry and an affiliate professor, member of the Biochemistry department here. And so Andrew had his undergraduate degree from Iowa, the University of Iowa. He then went to get his PhD at Johns Hopkins with a really wonderful enzymologist by the name of Craig Townsend. And he has one of probably the few people who has a PhD genuinely in biophysics from a biophysics department. There are very few of those around. He then went on to a postdoc with Francis Arnold at Caltech. And Francis Arnold, who won the Nobel Prize just a few years ago, has really pioneered approaches and the ability to evolve enzymes for specific industrial and biocatalytic applications. And he's contributed to that in her lab and in his own lab here. And he'll be telling us about some of his new stories with new enzymes. And so with that, take it away.
Okay, thank you so much, Aaron. Let's see, get the pointer going. All right. So first, let me say thank you, Aaron, for the invitation to talk. I feel like I'm a little loud here. And it is so much fun to get to share science with friends, especially doubly so in person. And also hello to all of you in the virtual world out there. It's nice to feel your presence as well. So I'm really happy to tell you about the work that I've started in my group here in Madison. I came here in the summer of 2017, and it's been a ride. My group, broadly speaking, uses enzymes to do organic synthesis, or at least that's what we want to do. That was the goal when we started. And it's worth acknowledging that this pursuit, using enzymes to make molecules, is not new. It has been around for a long, long time. One of the most, and it's used on all scales in industry. So here, if we think about commodity chemicals, on the left, the red's not going to do a lot for us, but on the left we've got a picture of BASF's plant for the production of acrylamide through the enzymatic hydrolysis of acrylonitrile. So it's done on multi-ton scale every year, and it's a good enzyme. We like it. So enzymes don't have to be used on such enormous scales where they are their own building in order to have an impact. So I found this nice picture of an industrial kind of medium, for them it'd be medium scale, kilogram scale production from a contract research corporation called France Evaporation that will conduct biocatalytic syntheses for other people, maybe for the synthesis of an active pharmaceutical ingredient, an API. So there's important uses of enzymes on this scale. But before you know maybe what molecule for a drug you want to scale up, you've got to scan around and maybe do some traditional medicinal chemistry. So here's a picture of a few grams of a molecule that could be used to be derivatized to probe the molecular properties on a much smaller scale. So here we can think of enzymes being used to make molecules just to explore molecular space on a milligram scale, and they go all the way up the pipeline to the largest scales in industrial catalysis. And starting off as a new group, as an academic lab, I recognize we're not going to do multi-ton scale catalysis. We really want to have an impact that we can have as a young, high hopes and dreams lab. So it's worth kind of pausing and recognizing, what are people already really good at? So among the transformations that biocatalysis has had the most success with are those that do functional group interconversion. So here's one recent paper from apologies, I don't have the date. This is from Pfizer. It's 2 years ago. They were producing a multi-kilo synthesis of a gamma secretase inhibitor, and they used enzymes to do this chemistry. In one case, they convert this ketone to a chiral amine. And they do this with a highly evolved aminotransferase enzyme. It's pretty neat. They actually use isopropylamine as the amine donor, and then acetone accounts for the mass balance, and they just blow that off, so that's kind of nifty. This reaction on the right is the reduction of alpha keto group to a secondary alcohol. And here they use a cofactor regeneration system, and this stuff is really successful. This happens on multi-kilo scale all the time, even though it's not really the enzymology that I learned when I was a biochemistry major at Iowa. The kinds of transformations, though, as one steps back and looks at the literature that are really missing, are the ones that put these molecules together in the first place. It's the CC bond-forming reactions. These are, we teach in our organic synthesis classes that this is, often the harder chemistry to do, and in fact, it still is today in biocatalysis. So my group was really focused on trying to find enzymes that were going to do potentially useful CC bond-forming reactions. But you still got to make something. There's got to be a product on the other end, and we don't want them to be some esoteric molecule. I would want a molecule that speaks to me, to my background as a biochemist, and maybe to you as well. So the molecules we picked are amino acids. These are arguably, I would argue, the premier building block in biological systems. So whether you recognize it or not, penicillin is actually made. It's a hyper-modified tripeptide. Vancomycin here is really tough to do in a chem-draw, so I just used a pymol structure. Vancomycin is a hyper-modified peptide. The most modified peptide that is known is actually polythionamide. This is ribosomally encoded and then subject to really intense post-translational modification, including methylation, hydroxylation, lots of L to D epimerizations, and life even goes to the trouble of putting a T-butyl group on one end. So really, really kind of high-end modifications to make this natural product. And medicinal chemists are savvy to this too, that by using amino acids and their analogs as precursors, we can rapidly access a large functional diversity in order to tune the bioactivity of molecules. But what medicinal chemists kind of tap into for their chemical diversity is still pretty sparse compared to the chemical diversity that nature is able to make. And that really leads to the mandate that I try to keep in mind for my group and the impact that I want our research to have kind of in the near and far term, I hope. That is, we want to develop enzymes to use biocatalysis to open new areas of chemical space for medicinal chemistry. We want to provide access, easy access, to more functionally rich non-standard amino acids that could be used as building blocks in medicinal chemistry. Now, I'm not going to get the chance today to talk about all the chemistry that's going on in my lab, so I wanted to just take a brief moment at the beginning to acknowledge my fantastic group that does all of the work that I'm going to talk about today. And we make amino acids of all shapes and sizes. They're very much our friends. And before I launch into a more detailed scientific story of trying to develop an enzyme as a kind of practical, scalable catalyst to make some non-standard amino acids, I want to pause and reflect, because I'm talking in a biochemistry department, that success in biocatalysis, the ability to use an enzyme for synthesis, is not going to use the same metrics that at least I learned when I was a biochemistry major to judge the qualities or the properties of an enzyme. So here I've got some data from the lab. The reaction isn't super important, so I've left it cartoony for well, it's actually a really cool reaction, but I've left it as a cartoon to just focus on the initial velocity period of this reaction. So here, we're watching A plus B come together and make C over time, and we can see as time progresses, the rate of the reaction slows down. And I learned as a biochemist that this is the useful region, this is the initial velocity region, this is where you're learning about the properties of the catalyst when the substrate concentration doesn't change a lot during the course of the reaction. Whereas for synthetic utility, that initial velocity region doesn't really tell you a lot. It's not enough to just start off making a product really fast. You want to know if it goes all the way to the end. So you're going to see me talk about the yield of a reaction, even though I'm talking about an enzyme. And in many cases, that's a more operable parameter than the catalytic efficiency measured under initial velocity conditions. You might also note that the substrate concentrations look like they have typos in them. We'll commonly work at tens to hundreds of millimolar substrate with biocatalysis because it's easier. I don't want something that's more dilute. You want to keep it nice and concentrated. So when we think of the classic Michaelis-Menten kinetic analysis, most of the substrate concentrations that are going to provide rich information about your Kcat over Km are way under the operating concentrations that we're going to use in upscaled reactions. So instead, for synthetic utility, Kcat kind of rises to the fore, and Km, sometimes it has an important role, sometimes it's just in the background. You just add more substrate. But even Kcat as a measure is not going to tell us everything we need to know about an enzyme. Again, that's often a reflection of speed under initial velocity conditions. But you want the reactions to go all the way to the end. So instead, what you'll also see in my talk is something called a total turnover number. And it's actually just what it sounds like. It's the number of times a catalyst does a reaction before it dies, basically. So you might see an enzyme might operate at high initial speeds, but then slow down. Maybe it's getting oxidized, maybe it's precipitating and unfolding, and the total turnover number is the aggregate, the integration of all of that information to show you how far can you push a catalyst in its reaction. Brief side note here that we're often not going to work under buffered saline conditions at pH 7.2. We'll just tune the pH to whatever makes the reaction work best as judged by higher total turnover number, and we're often adding co-solvents that aren't present in life. So you'll see a lot of DMSO or isopropanol in our reactions as well. And lastly, I'll say that for biocatalysis, we often don't want enzymes that are highly specific. That is, if we're going to go to the trouble of developing an enzyme to do a transformation, I don't want it to just do that one transformation. I want to do that transformation on lots of related substrates so that the catalyst is very promiscuous. And that's counter to what we often see as a virtue of enzyme chemistry, that enzyme chemistry is highly specific. So kind of altogether, I think of biocatalysis as a field that brings new perspectives to enzymology because we're asking different questions. We're asking to evaluate enzymes not for their properties in a native metabolic setting, but what they might do for us in a flask. So with this kind of background, and I hope for some of us helpful perspective on how we're going to be treating enzymes a little differently than we learn in our biochemistry textbooks, let's dive into some actual reactions. So non-standard amino acids, these really premier building blocks in biochemistry, are particularly difficult to make if they have extra stereocenters on them. For all the reasons you maybe remember from your sophomore organic chemistry, like figuring out which is R and S, and are they diastereomers or enantiomers? Well, the chemistry is that complicated too. And the addition of a single hydroxyl group is often a modification in natural products. So vancomycin features several of them. Fluorothreonine is a very simple natural product. It's one that Amy studied in graduate school. Where's Amy? Hey, what's up? I didn't even think about the connection. But here we are. So beta-hydroxy amino acids are very important. You can also kind of march down the backbone from alpha, beta to gamma, and there are natural products that have hydroxyl groups in the gamma position as well. So caspofungin is a clinically used topical antifungal compound. Alpha-manatin is the toxin from death cap mushroom. Clavalinine is, it's not clinically used, but has antibiotic properties. And I've highlighted here the gamma hydroxy core. And if you're really paying attention, you'll realize that caspofungin also has beta hydroxys. And it's in fact just basically made out of, and I guess that would include threonine, it's just made out of these modified hydroxylated amino acids. So we're going to talk about one enzyme system that we've developed, a threonine transaldolase. We're going to look at its mechanism, its structure, its scope to see how it performs as a catalyst to make these beta-hydroxy amino acids. And then we're going to talk about an enzyme that makes these gamma-hydroxy amino acids. And in this second story, we'll find that the native enzyme wasn't very good at it, and so we had to engineer it to get a lot better. And so you'll see directed evolution in that second portion. And then a common theme throughout both of these is that we don't want the enzyme to make just one thing. And we'll raise this question, and I hope begin to answer this question, of how we can control substrate specificity during directed evolution. So launching in. These threonine transaldolases were discovered about 20 years ago now from O'Hagan. And it was the biosynthesis of fluorothreonine where these enzymes were first found. They bind threonine. They use the pyridoxal phosphate, PLP cofactor, and they basically exchange the side chain of threonine, which releases acetaldehyde, and do some kind of addition into an alternative aldehyde. So you're just swapping, hence the name transaldolase. And this is used in fluorothreonine biosynthesis and albumycine. There weren't a lot of examples known. Most of the other beta-hydroxys in nature are installed through other types of mechanistic pathways. But what really drew my attention when I was starting my lab right around 2017 was the Vinchevitz lab published the biosynthesis of this obafluorine molecule. And it has a beta-hydroxy amino acid in its biosynthesis. And unlike the other threonine transaldolases that had been studied before, this one was well behaved. So they showed that you could express it in E. coli, which is not a given for all enzymes. But if you want to do a practical chemistry, you got to be able to get your hands on it. So Prashant and Tony were the ones who really led this effort in the lab. When we overexpress the enzyme, that's not a color defect. The E. coli turned pink, very bright pink. And when you purify the protein, you get a lot of protein from a liter of culture, 200 mgs from a liter of culture, more. And when you purify the protein, oh yeah, 250, it's on my notes. It's pink. OVH is pink, and that's not normal for a PLP-dependent enzyme. I usually think of them as yellow. And we can see spectroscopically a little bit of what's going on here. There's a broad absorbance band at 515 nanometers. And what Tony noticed one day when he was working in the lab was that, yeah, the protein was pink at the start of the day, but it seemed to lose a little bit of its pizzazz as the day went on. It just kind of fizzled out. And in fact, he went and stuck it under the Wisconsin sun and found that it photo-bleached. It went away. The color was gone, but it's not like it was destroyed. Actually, if you subject this to just green light irradiation right on that 550 nanometer peak, you actually increase the population of what looks like your kind of typical protonated Schiff's base, how PLP is typically bound to enzymes. So not only does it increase this population, but it actually makes catalyst that is faster, which is what you would expect for increasing the concentration of a catalytically active species. And so this was our jumping off point to do much cleaner spectroscopy with this protein. So we went ahead and added substrate. So this is a threonine transaldolase. Threonine is the first substrate that it binds. Threonine binds covalently, and it releases that active site lysine. And the side chain between C-alpha and C-beta is kind of poised, what's called periplanar to the pi system of the cofactor, such that it can do a retroaldol cleavage. And this forms what is basically a resonance stabilized carbanion that in the PLP literature is called a quininoid. And we can see these quininoids with their highly delocalized electrons have very intense absorbances right around 490 nanometers. So we can see the formation of the glycyl quininoid from OBH when threonine is added. And or at least I thought it was pretty amazing about this enzyme, is that it just seems to stick around forever. The half-life for the decay of this species is close to 3 hours, which on an enzyme, like on a biological timescale, when I think about macromolecules, that's bizarro. And it's not just stuck there. It is slowly turning over. It is getting protonated, which makes sense, right? Carbanions are basic. It gets protonated, it makes glycine, but if you add a lot of glycine, this isn't a thermodynamically stable species. You can't enter this in reverse. This is a persistent trapped intermediate in the enzyme active site. And if instead of just waiting around, you give substrate, the substrate will rapidly react in the mixing time of the experiment, and you'll see the quininoid population decay and formation of new adducts increase. So this is a genuinely high energy intermediate that is poised to react with different nucleophiles. So in kind of allied work here, we went ahead and tried to scale this catalyst up to see how it would work preparatively. So the simplicity is the name of the game in biocatalysis. You want to do, like if I'm going to ask you to make a molecule, I want you to do it the simplest way possible. Otherwise, you're not going to want to do what we recommend. So in this case, we're just using whole cells. Grow up E. coli, spin them down, and then add them to 1% weight by volume. That's arguably the easiest way to deliver catalysts. And so that's what it looks like here in one of these scaled up reactions. We add a fairly large excess of threonine, and then we just put them in the shaker overnight at 37°C. And we show that the enzyme reacts with a wide variety of benzaldehyde analogs. The absolute configuration that is favored by the enzyme is the same configuration as the natural product, obeflorin. So that's the major diastereomer in all of these reactions. The stereo control is pretty good, but in some cases it's not so hot. You get that with enzymes. What was really starting to be more interesting is instead of a simple benzaldehyde, if we had much more sterically demanding substrates, we're still able to isolate some compound with pretty respectable diastereoselectivity. And these types of heterocycles are really the cream of the crop for medicinal chemists. This is like their favorite playground, but it's been a tough place to access with good stereo control. So now they have access to these. Also, it's worth noting the enzyme works with aliphatic compounds, so it works with the gambit of reactive aldehydes. And we can even scale these reactions up. So here's beta-hydroxyleucine, which we've made like a gram of at this point. And these are useful molecules for biochemists that are interested in studying the like not just med chemists, but people that want to study the biosynthesis of natural products. Need authentic standards when the natural products they isolate have added stereocenters. So as soon as we posted this, it's published in Kim Biochem now, but as soon as we posted it, labs were emailing us to get a sample of our beta-hydroxyleucine because it's a known compound in the literature, but it's a hard ask to get an organic chemist to do 6-step synthesis for an acknowledgement in a paper. But we're happy to send it out. It's a pretty good catalyst. It's got a good scope, so we want to learn more about how it's working. So we worked with Craig Bingman here in biochemistry to solve the crystal structure of the protein. We got a pretty beautiful structure at high resolution. The crystals were very twinned. That's why the R factors look a little garbagey compared to what you might expect. But the structure itself is very clear. It's very clear, but it's kind of got a mystery to it. You see, this residue that's hydrogen bonding to the purity nitrogen is this glutamate 107. And that glutamate 107 is not conserved throughout divergent evolution with other members of the threonine transaldolase family. Instead, the residue that's conserved is this aspartate 204. So this really raised the question, which one of these is responsible for catalysis? And I'm not going to tell you the answer. This has all the makings of a great Agatha Christie novel. It's a real whodunit of enzymology. Is the crystal structure right? Is it the bioinformatics? And you are going to have to come to Prashant's defense in 2 days to find the answer. I had to figure out where to cut things, and I figured, you know what? This is a good story. I'll let Prashant tell it. And so he worked with John and Grace to answer this. Okay, so if we kind of take a step back from the work that we've done with OBH, I think there's 2 features that really struck us about the enzyme that help make it useful. The first is when we solved the crystal structure and did some subsequent modeling to try to get a sense of what the active site looked like, it's got a giant cleft for substrates to come in. And that makes a kind of intuitive sense to those of us that, to a biochemist, structural biologist, right? If you clamp around the substrate and you've got some perfect selectivity, you're going to be much more sensitive to whether or not you add a methyl group here or a whole phenyl group there. But this enzyme has an open active site. But just having an open active site is not enough for promiscuity. You need to be able to react. And this is where the long lifetime of that intermediate is, I think, a crucial parameter for what makes this enzyme synthetically tractable with diverse substrates, is that the other molecules that are reacting have all the time in the world to get in there and make a bond. because the decay pathway is like 3 hours here. Yeah, so this is what we've learned with OBH, and we're continuing to learn more of it. We're doing more engineering, and we can hear more about this from Prashant on Wednesday. So that's exciting. From the OBH world where we make beta-hydroxy amino acids, we'll move on. One modification, just kind of down the ring, if you want that hydroxyl group at gamma instead of beta. These are not really well understood in nature, I'll say, how these modifications are made. And so it was pretty exciting when, just a year before I started here, I was perusing Angavanta, and I saw a group was reporting the biosynthesis of this RIP natural product, eustaloxin, and they showed that this gamma hydroxy group is appended late in the biosynthesis from an aldehyde precursor. And what they show is that this enzyme USTD binds aspartate, forms some kind of nucleophilic enamine intermediate. So here we have an amine next to an alkene, enamine. We remember our grove, right? This is a good nucleophile that can add into different aldehydes to make gamma hydroxy amino acids. Or while in the first paper they show that it binds specifically to the corresponding aldehyde of the eustaloxin B precursor. How the enzyme works with other substrates, who knows? You can imagine this catalyst might have some real exquisite specificity for binding the natural substrate, and that's maybe when it knows to decarboxylate aspartate. That's how enzymes commonly work. But these gamma hydroxy amino acids, as prevalent as they are in natural products, are basically absent from the MedChemist toolkit. It is boutique, unique synthesis to make any one of these, and the ability to make lots of them with one catalyst in a straightforward manner would be very desirable. But almost nothing is known about this enzyme at this state. It's still in its egg stage. So what we do now is we go on a slightly longer journey with this enzyme. We're going to start off just looking at expression and activity. I told John I don't really like just listy outlines, and he really took that to heart when he made this one, I think. We're going to do some reaction condition optimization. That's relatively boring. Then we'll enter this Coupa castle and do some protein engineering. We'll look at the substrate scope. Did it get better or worse? Did we make a specialist? Let's find out. And then we'll scale up our reactions and go into Bowser's castle. So let us begin our little journey. So here we are expressing USTD. It's not like OBH that expresses at 250 mgs of protein from a liter of culture. We're happy 10 would be the best day on a wild type protein, which for a biochemist, that's a fine amount. You can get the job done with that. But if you want to scale things up, access to catalysts really matters. So we didn't have access to the native substrate, so we added benzaldehyde. Simple, non-enolizable aldehyde solution, and we were very pleasantly surprised that the enzyme did about 1,000 turnovers over the course of an overnight reaction. That's great. We didn't do any engineering, and it's already making something. We don't really know what it's making stereochemically here, but an undergrad in my lab, Eric Junis, who's since graduated, scaled up a reaction, crystallized the product, and we found the absolute configuration matches what was in the native natural product, which maybe isn't a surprise, but it sure is good to know when your molecule that you're testing is very different from what's found in the natural world. We went ahead and started some directed evolution here. We don't have a crystal structure of the protein. Very little is known about it. So we used a homology model to try to pick some residues that might be in the active site. Seems like a good place to start. And from 6 libraries that John screened, we found one that gave an activating mutation in the neighborhood of a 2-fold boost. In parallel with this, we figure, let's just try it nature's way and just do random mutation. So Prashant did global random mutagenesis, screened around 700 colonies, and found another mutation that gave in the neighborhood of a 2-fold boost. And if you're drinking your coffee and really awake, you might notice that these residues are neighbors, which was a total surprise. And this is the one hit we got from our library. So that's like a fun thing about directed evolution is you get surprised sometimes. So I think this naturally begs the question. These residues are predicted to be close to the active site. They're both making things a little bigger. You could imagine a scenario where it's like, well, this one's activating, put them together, more activating, right? Or you could think like, no, I understand how enzymes work. This is like, it's got a good fit here. It's like trying to put a key into a lock. And 2 keys could fit the same lock, but you can't put 2 keys in at the same time, right? Right? That's what I thought. So I told the students, whatever, go ahead and do the experiment anyway. It's going to be a big donut in terms of results. And I was wrong. The mutations are totally additive. And it still baffles me. And so since the protein seems to be able to tolerate variation here, let's go ahead and add more variation beyond these 2 mutations that we kind of stumbled into. So we did some 4-position degenerate codon mutagenesis, and we found 2 variants that I like to highlight, not because they're particularly better than the double variant that we landed on by adding these 2 together, but because these enzymes that have these FVF and TLM mutations are both activated, and they share no mutations in common. So there's no magic mutation to turn the protein on. There seems to be like a lot of ways to do it. What a pleasant surprise. So from our engineering land, we've powered up, and now we need to see if the evolution that we did, which is for a higher reaction with benzaldehyde, is going to translate for anything else. So Megan Campbell worked with John on the substrate scope of this enzyme. Isolating, purifying, and characterizing these is truly heinous. So all hats off to the team for working on this. So here we are under slightly different reaction conditions. We can see we're going to use total turnover number to measure how the catalyst is doing. And we see both variants are more active than wild type. TLM seems to be the kind of champion of the group. And what's shown in light here is the abundance of the opposite diastereomer. So the lighter bar that we've put on top is when the hydroxyl group is towards us, and the major diastereomer is the hydroxyl group in the back. So it's more active with benzaldehyde. That's what this indicates. And if we give different analogs, so different pair-substituted molecules, we see what in organic chemistry we would call Hammett-like trends. So if you add an electron withdrawing group, it's much more activating. If you add an electron donating group, activity seems to go down. But the variants are all more active than wild type, so that's a good sign. They're all at the para position, but we can look beyond that. So we can add other kinds of arenes, disubstituted molecules, and in almost all of the cases, these engineered variants are more active than wild type. Histidine, or this homocystidine histidine side chain maybe being an exception. These are all with various degrees of aromatic planar molecules. These enzymes are also able to turn over reactions with aliphatic substrates and even, I guess I've drawn the neutral form, but negatively charged substrates as well here, presumably the imidazole protonated. So this is really a promiscuous enzyme, and this was really lucky. We only engineered it to react with benzaldehyde, but we seem to get everything else along with it. We have our cake, and now we're going to eat it. So we're going to go from scope, now that was all at analytical scale, to upscaled reactions. And this is where protein concentration is a real bummer. 8.5 mg of liter means you're going to be doing protein preps every day to scale up reactions, and that's no way to live a life. When we look at USTD, the enzyme, the wild type one has 10 cysteine residues. It's like 400 and change amino acids. So 10 is a lot. And there's an old crystallographer's trick that if you have cysteines that are on the surface of a protein, under long time periods, they can tend to oxidize and form mixed disulfides. And those mixed disulfides crap out of solution, excuse me. And then that's why your reactivity goes down. That's why your proteins are insoluble. So instead, what you can do is just mutate all the cysteines that are predicted to be on the surface to alanine, and you'll get a more soluble protein. And we did that, and we killed our enzyme. Because it's not enough to be soluble, you need it to have all those good functional properties that we want as well. So we needed to be a little bit more savvy here, but we're protein engineers, so we'll just make different mutations. And I'm going to really simplify what was an elegant protein engineering effort that John made here, which was to sample a sparse library of different mutations for 5 different surface cysteines, sequence which variants we were able to measure activity from, and build a really simple linear regression model to predict which variant would be most active and have the fewest number of cysteines possible. So we were able to mutate out 4 cysteines. This left 1 behind, so we call this enzyme USTD 2.0. And lo and behold, it expresses, we get like 5 times as much catalyst. And now when we use our preferred simple whole cell mode of catalysis, just adding 1% weight by volume whole cells lets us drive reactions up into a range that would be synthetically useful. So this was exciting. The scale-ups, still not trivial. We often had to do a lot of protecting group chemistry, which I feel like is really sad for a biocatalysis group, because you want to say, like, oh, it's protecting group free, but you still have to isolate and characterize the molecules, and that meant making them greasy. So we get reasonable yields for these reactions. And even, remember I said that mutating those surface cysteines increases crystallizability? It increased crystallizability, which was real nice. And I will say, we had crystals of this that we shot so many of with Craig for like a year. And we got, at the very best, like 2.7 angstrom resolution, but we were never able to phase the thing. And it was hurting. It was hurting. And then we left. We weren't able to do anything because it was 2 years ago. And the trays that Craig had harvested crystals from just sat in the back of the crystallography lab, and the ones that entered the pandemic with clear drops came out of the pandemic at some point with big, beautiful crystals. And then all of a sudden, we were able to see what was going on here. We got much higher quality crystals. We got a structure at 2.25 angstrom resolution, and this let us see into the active site and find that trio of mutations that we had put in early in the evolution to see how they were changing the active site. So this work's been accepted. That's really exciting. I think I'm doing okay on time. I'll have to speed up a little bit here. To get to the final end, which was, and I tried to be really honest with the evolution of USTD. We'd evolved it to react at all of the stages with just benzaldehyde as our model electrophile, and we got lucky. But we don't always get lucky when we're doing directed evolution in the lab with these kinds of model substrates. So one project that we started very early in my group was on the decarboxylation of substituted tryptophans. That would make a corresponding tryptamine. And the tryptamines, even if you haven't heard of tryptamine before, I'm sure you're familiar with its better known serotonin, 5-hydroxytryptamine. You can make simple modifications of the indole core, that's the name of this 5-6 fused ring, to make more selective agonists of different serotonin receptors in the cell. And then other kinds of modifications lead to yet different types of inhibitors. So it's we would say, a pharmacologically rich space to make these tryptamines. And we wanted our enzyme to be a really good catalyst at decarboxylating these tryptophans to make the corresponding tryptamine. And looking here again with turnovers, we see the wild-type enzyme can perform tens of thousands of turnovers before it dies. But as soon as we put a substituent on the 5 position, it seems to just kill it. And somebody else had solved the crystal structure of this. We built a little model, we looked, and lo and behold, if you model in like where a 5-chloro group would go, it's just got this really nasty steric clash. So I was like, okay, I think you know what to do, right? We're going to mutate, we're going to make it better, and so we're going to pick a model substrate. We're going to use 5-methoxytryptophan, which previously here, right, this is kind of the poorest substrate that we could measure a signal with. And we're going to use this as our proxy for 5 substituted materials. So we've got our 5-methoxytryptamine, and we're going to screen a library at W349. We're going to take the biggest residue, we're going to mutate it, and lo and behold, basically everything is better. So, protein engineering, it works. And I'll say the mutations are kind of surprising. It's not like, I would have guessed maybe methionine, keep it long and dangly, but a little greasy. But no, it's kind of all over the place with this. But that was just activity on 5-methoxytryptophan. And this is typically how directed evolution goes. You pick a model substrate, you screen your product formation. I'll say we do all of the product screening by LC-MS in my lab because they all fly on MS in positive mode. And you maybe got some parent, you get a most activated variant, you go through maybe multiple rounds of directed evolution, maybe just one if you thought you had a good one, and then you're going to check what happens with other substrates that were not part of your selective pressure. And we don't really know anything about the scope until we test it. And when we tested the reactivity of this new W349K variant, yes, it's better on 5-methoxytryptophan, but it is worse on basically everything else that we threw at it. Bummer. And if we want to make lots of different substituted tryptophans, what are we going to have to do? Engineer a new variant for every possible reaction? That's way too much work. 10 years not that long, right? Plus, it's boring. Owen would have walked away if I pitched that. So we realized we just needed a different approach. And we're searching by mass spec. Oh, and let me pause here and say that this strategy of picking a model substrate for your evolution, this is how directed evolution was originally done. So this is the first directed evolution paper. It's like totally readable, highly recommend it. This is how directed evolution was done when I came to the Arnold lab and I finally learned how to do directed evolution. It was a lot of fun, worked on one substrate. This is how industry does directed evolution. This is how basically everybody does it for biocatalysis, where you want to scale it up and do single substrate reactions. And this is kind of breaking the first law of protein engineering in some ways, that you get what you screen for. What we want is a generalist that will react with many substrates, even though a single substrate is the standard. So I think you see where I'm going with this. What we need to do is invert things. Take that scope information and put it early in our screening step. That way we can use our mass spec, where as long as we pick substrates that have different M over Zs, we'll be able to resolve them uniquely without any really special separations or anything. The instrument does all the work. Then we're going to get two kinds of information out of a library. Instead of just total activity, which I'm going to represent as these diamonds in the middle of a plot, they're all going to be rank ordered from high to low, so you kind of know what to look at. We'll also get information on the relative abundance of the different products. That's what's represented in the different colored bars here, corresponding to maybe different substrates reacting in different ratios. And plausibly, you could have variants that have lower activity overall, but seem to shift to be really good at one substrate. So it's more information rich in theory. In practice, we'll go to the literature, see what other people have done, then we'll go to the lab and try it ourselves. So in the literature, these types of mixtures to assess enzymes have been around, these types of multiplexing methods, they've been around for a while. They were called fingerprinting by the Raymond lab. And here they didn't, if you read it, you realize they're not actually multiplexing. They're just doing a lot of reactions in highly parallelized fashion and then using that information altogether to make decisions. But then a few years later, they realized that they could put all the substrates in one mix, and as long as they're in that crucial core to the enzymologist initial velocity regime, they're going to get good information on substrate specificity with their catalyst. So fingerprinting as a term has been used for both, so we need to use something else to be really clear that we're referring to a mixture, and a mixture that's not necessarily going to be under initial velocity conditions. So let's take a look at what happens when substrates are mixed head-to-head here. So we're going to look at our TDC, our tryptophan decarboxylase, with its native substrate, 2 analogs. When we put in a mixture, we can quantitate the amount of product that's formed using standard curves for all the products, and we see that the product distribution is qualitatively similar to those total turnover numbers, the numbers that often tell us how useful a catalyst will be. But it's not qualitatively the same, or it's not quantitatively the same. But I'm talking to a bunch of biochemists, so I think some of you in the audience might know the answer. What actually determines the ratio of the products is the ratio of the catalytic efficiencies under these conditions for unimolecular reactions. And here, when we measure the individual kcat over kms for the substrates, and we compare them to their percentages in competition, it is a bang-on match. And the only error here is basically the error in the measurements. So this has been known for a long time, that multiplex methods provide the true readout of specificity for an enzyme, and these have been used by protein engineers these days as well. So in practice, though, not just under initial velocity conditions, other factors might influence what you get if you're screening on a mixture of substrates and not just one pure substance. So here, if we can envision adding to our mix a 2-methyltryptophan, this TDC is not very good with it. In fact, at our early 1-minute time point here, where we've got a pretty good reaction going with 6-chloro and the native substrate, we can't even see the 2-methyl. But if we wait a lot longer, we'll lose information about what's happening in the initial velocity regime, but we'll gain information about the promiscuity of the catalyst, how it reacts with poorer substrates. And I'll say that there isn't a right or wrong answer here. The point is that we have a choice for what we want. And for often the pursuits in my lab, activity, as it shows on the right-hand side here, is going to be more useful than something that's strictly under initial velocity conditions. So let's test this on a library. Let's go back to our wild-type tryptophan decarboxylase. Here we've got a mixture of substrates. We're going to mutate that same tryptophan that we targeted earlier, and we can now see, with the same amount of screening effort, it's all by LC, we see how all of these mutations shift both the total activity and the specificity of the catalyst, or promiscuity of the catalyst. So blue was the substrate we had screened on before, and we see that actually this tyrosine, W to tyrosine mutation is probably a better generalist than the W to K variant that we had initially been drawn to. And we didn't have to do any more work to get these data than we did the first time. So this approach seemed like it was successful, so we're going to apply it all over the enzyme active site. The 4 residues that are in light pink, we never got any good hits for these libraries, so I'm not going to show them to you. And we seem to get pretty significant increases in activity with substrates from the residues that are in magenta in the enzyme active site. And so we're going to now validate in single substrate what looked to be the best mutant from these competition substrate multiplex screens. And it works. It's great, right? Because it's not really a given. Activity in a mixture might not be the same as activity on a pure substance. With every single substrate that we had in the screen, we were able to find a variant that had increased activity. And it wasn't always obvious where you would go to find an activating mutation in the active site. I really get a kick out of the activity with 4-bromotryptophan. Here, you would expect for something that's projecting in this direction, a mutation to a smaller residue like leucine to alanine would be like the way to go. And that is an activating mutation, but mutating it to the large floppy methionine seems to be even better. And this is something we learn time and time again in protein engineering. You get kind of surprised by what's good, and you never really know unless you test. So in this case, we were able to do this classic protein engineering effort, boost activity with analogs with no rescreening of the libraries. And many of these activating mutations aren't what you would have predicted. So that's pretty fun. This is all fairly straightforward. This relationship between specificity and the underlying kinetic constants holds for unimolecular reactions, but with a bimolecular reaction, which could be more kinetically complex, this type of relationship might not give you a clean absolute readout of specificity. So it might get a little bit dicey. So what other enzyme could we test this method with to see how it works? And we're going to use the enzyme that's making all those tryptophans. Instead of a unimolecular reaction, it's bimolecular. Serine and indole come together to make a substituted tryptophan. This is an enzyme for which there's significant commercial interest these days. This enzyme I had evolved when I was a postdoc previously to work. This is going to be trypsinthase and just the beta subunit. I previously engineered it to work without its allosteric partner present. And it works on a lot of different substrates, but all that engineering was done at 75C. And for some applications, like if we want to put the tryptophan synthase together with the decarboxylase in one pot, that decarboxylase isn't thermostable. So we need a catalyst with higher activity at lower temperatures and hopefully maintain that broad promiscuity that drew us to this catalyst in the first place. So this is work that Peyton took on in the lab. So we're going to use a mixture of substrates. Now here we're going to use a mixture of different indoles. So here we see each different substituent gives a different mass signature so that we can resolve the products using the mass spec. We're going to start off with an enzyme that had been previously engineered. And I'll call your attention now that one of the substrates, this is called indoline when you've saturated across C2, C3 of the indole ring. This is no longer a carbon nucleophile. This is a nitrogen nucleophile. We just thought it'd be fun to put a nitrogen nucleophile in with the mix too. In hindsight, Good decision. So let's see how this sums works, not with a targeted active site, but let's see how it does with just totally global random mutagenesis. And so we're going to do error-prone PCR. We aim to get about 1 to 2 mutants per variant, and let's see how it does. Here's our parent enzyme. And when we make mutations, we can see that there's a large increase in the total activity. And because this substrate multiplexing gives us information on the product profile, we can see that all of these mutations look like they're going to be good generalist activating mutations. So this is great. We'd found these types of mutations before with TRPB, and some can do this as well. So that was nice. None of these mutations are close to the active site. They're all far away. That's kind of fun too. We also saw something else that was real interesting. We saw this variant, and at the time we didn't know what it was. It's just random mutagenesis. We had a whole lot of things on the plate that appeared to give a 3-fold boost in the amount of this dihydroisotryptophan, that product of a C-in bond forming reaction, even though the variant was way worse at doing everything else. I see I'm going to run a couple minutes late, but I'll try to speed up here. When we went to validate this reaction, indeed, the enzyme is a lot worse at everything else, but it also seems to be really bad at making DIT as well, even though it looked like it made more on the left. So this seemed like there was a conundrum. And what we found through kinetic analysis of the enzyme is that, and I'll abbreviate, each substrate in competition is a competitive inhibitor for every other substrate in solution. They're all trying to get into the same active site. And if you make a mutation that makes what was a really good substrate suddenly really bad, it stops being a good inhibitor as well. And a substrate that had a tough time getting the active site now gets a lot more dwell time. And so it'll actually have an increased amount of product formation in competition, even though it's lower in single substrate. So it's not like a false positive or anything. It's a reproducible, understandable kinetic phenomena. It's how enzymes work. So this information we can use in a kind of interesting way. We know that this mutation is talking to the active site because it's changing specificity. And maybe H to R here is just sending the wrong message to the active site. Maybe we can change it to something else. So we did that. We did a site saturation mutagenesis library, and we found that even though our intermediate had really low activity, we were able to leverage that information to find a variant with a much higher activity. And that was very exciting. It's always a good day when that works. The new enzyme is more active with many substrates, not just the ones under selective pressure. So wrapping up, what have we learned about this multiplexing method? That under ideal circumstances, product ratios are proportional to catalytic efficiencies. We're biochemists. We already knew this, but it's helpful to have a reminder from time to time that active site mutagenesis can track specificity shifts during the screening steps before we get any further, and that the hits that you find from competition can be predictive of activity with single substrates. So it speeds up a process and bypasses the need to rescreen libraries over and over, and that the variants that have lower activity can also be useful, immediately useful, to tell you which mutations are talking to the active site. This enables a kind of new step in a fitness landscape. So traditionally, when I think of a fitness landscape for directed evolution here, higher means more activity, and we know that you can find some activating mutations, but most ways lead back down the mountain. And occasionally there's so-called neutral mutations that might open up new paths towards the top. When we do screening in multiplex, this takes on a different view. We can do the traditional march straight up the landscape type of screening, but we also find out that many mutations that look neutral or deactivating are in fact not simply killing the catalyst. They're changing the specificity, and we can use that intermediate to make kinds of lateral steps or even go down before we choose to go back up again in a fitness landscape. So this is exciting. So here we can use specificity to change engineering. So sums give us more information from every variant that we've measured, and skip here if you guys want to talk about it more. And this is also practically useful as well because in medicinal chemistry they need reactivity with a broad scope. I also, since I'm talking locally, wanted to acknowledge how much fun it is to do science with collaborators here on campus. We've had a nice collaboration with the Cavagnero group who needed access to an isotopically substituted tryptophan, very specifically substituted, and we were able to make it with enzymes. We've also had a collaboration with Judith Burston's lab doing the first new-to-nature de novo biosynthesis of a cofactor, and it turned out E. coli could already do it. You just had to ask them real nice. So this is my group. I don't get to say this enough to them, but thank you. You are wonderful. You make this job good every day, and I'm so grateful to get to work with you. And we've got more stuff to do, too. All right, I've got great collaborators, and thank you all for your attention and patience today.
Thanks, Andrew, for that great talk. See if there's any questions. I'll start off with one, maybe a little bit philosophical. This one. So what limits the total turnover number for an enzyme?
Oh, lots of so what limits total turnover number? Lots of things. Basically, everything can limit total turnover number. So you could imagine a reaction is really fast in the beginning, but is product inhibited. So even though it could turn over more and the enzyme's still in solution, product binds. So that might not be common under metabolic settings where a subsequent enzyme can react with a product, but when you're in a flask, suddenly these effects become prominent. Or it could just be lifetime of the catalyst. More thermostable catalyst persists longer, can do a reaction longer. And also speed is good. Speed is always going to be good.
Yeah, I was specifically thinking of the middle possibility where the catalyst has a defined lifetime. Is it possible to evolve things which last longer?
Sure. Generally, more thermostable variants will persist longer in solution. It's thought, at least this is how the picture is in my mind, that you get kind of micro unfolding events and then things start to precipitate and crash out of solution. So the more stable your protein, less susceptible it is to these things. I think that's generally held to be the case in protein engineering.
Anyone else?
When you guys pick up on when you guys decided reaction conditions, do you try to make stuff like similar to the way we found industrially with those like DMSO or alcohol solvents, or do you guys switch to like stuff more traditionally used as solvents?
Yeah, so how do we pick our reaction conditions? That's a great question. In industry, the perspective is you figure out what reaction conditions will be optimal for your process, and then just engineer your enzyme to work under those conditions. We aren't working under industrial process scale, so I'll say we do what kind of works with as simple as possible. We try to avoid halogenated solvents. We also try to avoid DMSO because you can't rotovap it off at the end of a reaction, although gosh, it sure is a nice friendly solvent for a lot of enzymes, so that can be tough. Hey, Sylvia.
Very nice.
Thank you.
I was just wondering if you have any target that can be perhaps incorporated into some practical product or potential drug. So you think more in from the bottom up.
Yeah, so the question is there a specific natural product that we could make maybe more efficiently here? Probably, but as you know, appearances to the contrary, I'm not much of an organic chemist. So doing this type of research would be really hard in my group because I'm not really well equipped to advise students in organic chemistry. So we're more bottom up. But I will say, because what we're making are amino acids, it doesn't take a lot of creativity for other chemists to see how these transformations could be useful to them.
Did you?
Oh, yeah,
Yep.
I wanted to ask on the
Hi.
beta hydroxy project, why did the DR go up so much after you isolated them?
Oh, why does DR go up when you isolate? There's a natural tendency to hunt for the bigger peak. And so whether you're trying to or not, the DR tends to go up as you do a purification because, diastereomers don't necessarily have the same physical properties. That's why we also report the DR prior to any manipulations, the analytical ones, and that will be typically a lot lower.
Andy, I have two questions. Hi, about the 10-cysteine enzyme. The
Yeah.
first one is, did you consider doing your mutations to serine instead, sort of isosteric and maybe keeping more of the solubility?
If we had done that, that probably would have that likely would have worked a lot better. But we ended up just making a library instead. I mean, it's not any more work to clone a library than it is an individual protein. And we figured since we're doing a bunch of engineering for the story anyway, let's just add a little more on the mix.
Oh, I thought you said you tried alanine.
We did do alanine.
okay.
We could have also done serines.
Yeah, gotcha, okay.
Yeah.
Then my other question has to do with the crystal structure. And,
Sure.
you know, usually when it takes that long to get a beautiful big crystal, it's because, not because crystallization takes that long, but because something else is happening in the meantime. So what did you find in the structure? Were the cysteines oxidized eventually? Or what was interesting in the high-resolution structure that told you what made it crystallize over COVID?
So I don't know what made it crystallize. There were cysteines that oxidized, because that happens. Pretty cleanly, too. There are some residues at the crystal packing interface that look to be like two negative charges have to be real friendly with each other in order for the interface. So I think it's just not the greatest crystal packing. They also tend to be, the packing is also a lot better in 2 dimensions than in the 3rd dimension of the crystals, and that also contributes to poor quality. John is doing more engineering now to try to increase the crystallizability and maybe the solubility of the catalyst as well, because we always want more protein. Yeah?
Amy?
Great talk, Andy. With your sums of protein, what is sort of the limitation on how many substrates you can multiply? So just the availability of building blocks?
Yeah, so how many substrates can you add? And I mean, as many as you can measure or as many as you want to try to keep track of. So we found that quality, for what we do, where there isn't an obvious substrate mix, like for a protease working in vivo, you'd probably want to check the 20 amino acids. But for us, it's less obvious. So we try to pick quality substrates that have very different properties. And that seems to work just as well as measuring lots of things that tend to be highly correlated with each other. But that's an open question that we're trying to figure out. What determines a good mixture of substrates? So we're doing we're trying to figure this out.
So I'll have a final question.
Sure.
So have you gone back through the databases and seen if any naturally occurring enzymes happen to have the substitutions that you've evolved? Like perhaps in an organism that hasn't been cultured or
So
something?
I haven't gone back recently. With USCD, the enzyme that I showed most of the directed evolution with, there are very few examples of that sequence. It's an enzyme that's only known in the biosynthesis of a small set of natural products. So it's not like a primary metabolic enzyme where you have a really rich sequence record. And in the case of the trip decarboxylases, I'm not sure. I'll have to ask Alman that afterwards.
Great.
Yeah.
So let's thank Andrew for the great talk.
Thanks, friends.